All right, and now that we have gone over this overview, this quick one, now let's get back to the paper and now look at the paper's methods. And the first part is on detecting, quote, viruses. So now let's get back to the paper and take a look at the paper's methods. The first of which involves trying to, quote, detect known viruses. So methods, viral diagnostic methods. So 4 lower respiratory tract samples, including bronchoalveolar-lavage fluid, were collected from patients with pneumonia of unknown cause who were identified in Wuhan on December 21st, 2019, or later, and had been present at the Huanan seafood market close to the time of their clinical presentation. And 7 bronchoalveolar-lavage fluid specimens were collected from patients in Beijing hospitals with pneumonia of known cause to serve as control samples. Again, this paper doesn't actually go through much more information on these supposedly controlled samples. So the lower respiratory tract is the lower part of the groups of organs involved in moving oxygen in and out of the body. And the bronchoalveolar-lavage is a method in which a tube is inserted into the lower respiratory system and fluid introduced, then collected for examination. So if you have this right here, so there's a lower respiratory tract. So you put a tube inside, and then you pump in some water inside there, all the way inside, and then you would extract this fluid out. And that's the bronchoalveolar-lavage fluid there. So you pump it all the way inside here, and then you get the fluid out. Just a pretty invasive technique there. Continuing further. So let's continue further in the paper. So extraction of nucleic acids from clinical samples, including uninfected cultures that served as negative controls. Again, there's very little info on these supposed negative controls in this paper. So was performed with a high pure viral nucleic acid kit as described by the manufacturer Roche. And I'm not going to go over it though, but just the results or supposed results of. So this were cultures right here, so including unaffected cultures. So they would extract these from these supposed clinical samples. So a cell culture is a process of growing cells under controlled conditions, and a negative control is a condition expected to produce a negative result, such as giving samples without the active ingredient being tested. And here's a cell culture plate or quote petri dish, like that, just a plastic or glass case like that. Dish, I mean. And now here's epithelial cells in culture stained for keratin, which is a structural protein in red, and DNA in green. So these epithelial cells like that again. Hard to get too much detail on this, but it's interesting nonetheless. So continuing further, so extracted nucleic acid samples were tested for viruses and bacteria by Polymerase Chain reaction or PCR using the RespiFinder Smart 22 kit or Pathfinder BV and the LightCycler 480 real-time PCR system in accordance with manufacturer instructions, and samples were analyzed for 22 pathogens, 18 viruses, and 4 bacteria as detailed in the supplementary appendix. And this appendix just gives you the sequences or the list of these supposed pathogens and viruses that they were looking for, and bacteria. So the polymerase chain reaction, or PCR, is a method of rapidly making millions to billions of complete or partial copies of a specific sample of DNA. And PCR cycles refer to the repeating number of steps during DNA replication. So if you have a DNA template with sequence of interest, so you have this like this, a sequence, and then what you also have is a whole bunch of these supposed primers, or these starting sequences, or these targeted ones. Then this this dNTP or deoxyribose nucleoside triphosphate, and dNTPs are the building blocks of DNA that are used by DNA polymerase enzymes to synthesize a new DNA strand. So these are the building blocks of the DNA. A whole bunch of these compounds, and you have the polymerase, which is the enzyme that builds those up or makes the DNA. You have these primers, or these starting sequences, and so on. So denaturation. So if you have this target DNA template, denature it, you'll just separate these. So it usually heats them up, separates it, and then this is annealing. So the next step is that this target primer connects to them. This connects here, and then these are the building blocks of it. With this polymerase starts adding these, and then you can form a template like this, and I believe that they'll be matching up. That would match up like this, following this primer set. This should be a target of that. Yes, I'm not going to go into too much detail of that. And then we'll do it on this in this case, because you separate each strand. And now this builds it up using these building blocks and that primer initial setup. And then you do the same thing again. Then you would second cycle, you're going to denature, and then anneal, and so on. So each time you get double. Then the third cycle you double, and then the nth, and then it goes to copies are 2 to the power of n. You get a PCR product, and so on. So you basically amplify this to millions and billions of copies, and so on. So deoxyribose. Yeah, actually we went over those. So now let's continue further. And here, actually, before we go further, I just want to add this primer definition. So a primer is a short, single-stranded nucleic acid used by all living organisms in the initiation of DNA synthesis. Interesting. So that's the first one, and then it's gonna synthesize the remainder. So that's the start, and then it gets all these and matches up according to this sequence, or the likely sequence that it wants to get to. It's very interesting. So now let's continue further. So in addition, unbiased, high-throughput sequencing described previously. It was used to discover microbial sequences not identified by the means described above, and a real-time reverse transcription PCR or RT-PCR assay was used to detect viral RNA by targeting a consensus RdRp region of pan, this beta CoV, as described in the supplementary appendix. And let's go over some more technical terms. So high throughput sequencing, or second generation sequencing, or next generation sequencing, NGS, refers to advancements in sequencing technology that allows for entire genomes to be sequenced at once. And the term unbiased refers to the method where specific sequences are not preferentially amplified or sequenced over others. And here's just the history of sequencing technology. There's Sanger method, human mitochondrial genome sequence, 1981, human genome project, 1990, then 1985, complete cell genome, then 1986, complete eukaryotic genome. I think that's multicellular genomes. And then 2001, complete the human genome project, and then second generation sequencer right here. 2005, then second generation sequencer, genetic analyzer 2. So these ones do basically in bulk. And there's a research human microbiome project. There's all the microorganisms in the body. And then there's a third generation sequencer, PacBio-RS. There's a nanospace sequencing. And there's a third stage human microbiome project. Again, this is getting pretty advanced stuff. So real-time polymerase chain reaction, or real-time PCR or qPCR monitors the replication of a targeted DNA molecule during the PCR in real time and not at its end as in conventional PCR. And reverse transcription PCR or RT-PCR combines the reverse transcription of RNA into DNA, and then it replicates the DNA targets using PCR, which is, again, split the strands, and then you would then replicate and split and replicate. And reverse transcription is the process in which the enzyme reverse transcriptase is used to generate DNA from an RNA template. In an assay over here, because it's using this RT-PCR assay. An assay is an investigative procedure to detect or measure a specific biological entity or function. Basically, just a procedure list. And then the last one right here, so they use this RT-PCR to detect viral RNA by targeting a consensus RdRp region of this pan-B-CoV. Let's see what those all mean. So pan-beta-CoV refers to a group it refers to a group or test designed to detect a broad range of beta coronaviruses. And RNA right here, this RdRp, R is just stands for RNA-dependent RNA polymerase. RdRp, or RNA replicase, is an enzyme that catalyzes the replication of RNA from an RNA template. So this replication is RNA-dependent RNA polymerase. I believe we saw that in the over here. So this is the central dogma of molecular biology, and this is the RNA-dependent, so RNA polymerase, RdRp. So you need that enzyme to go in replication, either go from here or going back, and so on. Different sense. All right, that's very fascinating stuff. All right, so that is the so RdRp is the replicase, is the enzyme that catalyzes the replication of RNA from RNA template. And the RdRp region refers to a specific region of the genome of RNA viruses that encodes for this enzyme, this RdRp enzyme. And the consensus right here, so it's targeting a consensus RdRp region of these coronaviruses. And then so the consensus RdRp region refers to a segment of the RdRp gene that is highly conserved among different quote strains or isolates of a quote virus species. All right, it's very, very fascinating stuff. So now let's take a look at a summary of detecting the quote virus. So let's quickly summarize the quote virus detection methodology in this paper. So 4 samples were taken from patients with pneumonia of unknown cause in Wuhan on December 21, 2019 or later. And 7 samples were taken from patients with pneumonia of known cause in Beijing to serve as control samples. And nucleic acids were extracted from the clinical samples and, quote, uninfected cultures. PCR or DNA replication tests were done on the extracted nucleic acids to test for 22 quote viruses and 4 bacteria. And here's one to add this samples right here. So this is a lower respiratory samples. Yes, so the 4 respiratory samples. Same with this one here. 4 lower respiratory samples. And I believe this is the fluid. We're going back here. So 4 viral diagnostic methods. So 4 lower respiratory tract samples, including Bronchoalveolar or Bronchoalveolar-lavage fluid, and so that's including that. I don't know what it means. Is it all of them that or different? Then while this one right here, 7 this fluid samples right there for the known cause of pneumonia. So I'm just going to say. I'm going to assume 4 low respiratory fluid samples. And then the same one right here. Fluid samples were taken from patients with pneumonia and so on. So anyways, and then they were tested for these quote viruses. And I would assume that the known cause ones didn't, they would test for these, or at least some of these that they're being tested for. But then the unknown one wouldn't be testing for these known viruses and bacteria. So high-throughput sequencing were used to discover microbial sequences, looking for these molecules and not the actual quote, virus. Not identifiable by the above means, which we're testing for. Testing for the known sequences. And also, using PCR and so on. And going further, so RT-PCR, so again, this is, the PCR is DNA replication. Then RT-PCR is RNA conversion to DNA, then DNA replication. Assay was used to detect, quote, viral RNA by targeting a consensus RdRp region of a group of beta coronaviruses. And this is the genera of coronaviruses that includes the alleged COVID-19, quote, virus titled SARS-CoV-2.